All right, welcome back. We're going to continue talking about gut development. I've got a visitor, a doggo's here because it looks so dark in here, and like the colors are all bright, and my camera's having a hard time focusing because it's storming outside. It is 12:30, and it is dark. So you may hear my dog crying a little bit because she does not like thunderstorms. All right, so let's continue on with our gut development. We talked a bit about how the AIP and the CIP kind of pinch off, and we get the formation of the foregut, the hindgut, and the midgut. But we're going to look on the anterior side now and talk a little bit more about what happens, because obviously, and we talked a little bit, at least briefly mentioned offhand, the formation of the rectum at the posterior end. But on the anterior end, we have the beginning of our digestive tract. The gut is our mouth, right? So this is the formation of like the pharynx up at the top, and this arises from the pharyngeal pouches. Ooh, she's pushing me around. She is freaked out. So the pouches are symmetrical. So in this image here, we have the four pouches. So we have the pharyngeal arches, and then the pouches. So we have pouch 1, pouch 2, pouch 3, and pouch 4. And then again, this is split in half, so you'll have it on the other side of your face as well, or what will be your face. So in pharyngeal pouch 2 here, you get what becomes the tonsil, and so we can see this further on in development. In pouch 3 here, we get the parathyroid formation, or primordia, what will eventually be the parathyroid, and the thalamus, or thymus, excuse me, which is down here. We have the parathyroid primordia right up here, and this postbrachial body as well. So again, this is on both sides. In between, you can see that we start to form this auditory channel. So, you get a head cold. There's that channel between your inner ear and your nasal here, where you get postnasal drip and all that stuff, etc. Okay, now let me consult my notes to make sure I've covered everything. So the end of each of the first pharyngeal pouches, so on your left hand and your right hand side, those become the tympanic cavity in the middle ear and the eustachian tube. So we mentioned that. The second pouches receives aggregates of lymphoid tissue, and that becomes the tonsils, part of your lymphatic system, right? So when you get sick, they swell, they play a role in your lymphatic system. The dorsal portion of the third here, this green one, forms the parathyroid gland, and the ventral portion becomes the thymus. We got that. And they both will migrate caudally and meet with the tissue from the fourth pharyngeal pouch. So they'll kind of migrate down. You can kind of see right here, that's what these arrows indicate, is that tissue will migrate. So we get kind of one big clump then down here of the postthyroid glands, the thymus, the thyroid, etc. Okay. All right, so that's the pharynx. So just very briefly, pharynx, I almost put in one video, but it's only one slide with the pharyngeal pouches and stuff. So it got combined with the rest of this gut development. So this is kind of the regional specification of the gut endoderm and the splanchnic mesoderm. And this happens through reciprocal interactions. So reciprocal being in the news a lot lately when this video was recorded due to tariffs, meaning one influence, they bounce off each other, right? So if one does to the other does to one, right? Back and forth. So in this case, SOC 17, which is not, let's see, is not labeled on here, I don't think. So SOC 17, it helps to specify the digestive tube, so what is becoming the actual digestive tube, but it doesn't provide polarity, right? So it doesn't tell you, like, it says it's common to all the digestive tube, but it doesn't say what part it's going to be, if it's going to be stomach or intestine or etc., right? So the stomach develops as a dilated, so expanded region of the gut closest to the pharynx. And then as you can see here, we have C-SOX2, which is specifically expressed in the esophagus and the stomach. As you move further down the digestive tract here, we would have the small intestine. So PDX1 and CDXC are what start to specify the small intestine. HOX here is specifically for the branching off of the liver and the pancreas, which we'll talk about in another video, the specifics of the liver and the pancreas, because there's also influence here from the heart and aortic artery and the vitelline veins and things like that. So small intestine, CDXC, and then we get down further, CDXA is expressed. So this all provides the polarity is these. This is kind of the mapping, the gradient. CDXA for the colon, large intestine, the rectum, etc. Just making sure I've got everything. I think I'm good. All right. So here we have the regional specification of the gut endoderm and the splanchnic mesoderm through this reciprocal interaction. So we're going to get into more detail on it. So the differentiation from the splanchnic mesoderm and the surrounding epithelium cells. So here we have the intestine epithelium and then the stomach epithelium, right? And so what causes intestine and what causes stomach? So as this digestive tube is proceeding through the body or the embryo, like as it's not moving but like at different regions throughout the embryo, it will encounter different mesenchymes. And so these mesenchymes or mesenchymal cells instructs the endoderm to differentiate into different tissues. So we kind of covered that previously, right? So that's the basis of this, is that as this is proceeding from the top, from your head down to your rectum, this digestive tube is encountering all sorts of different gradients and different mesenchyme cells surrounding it, right? And those paracrine factors are influencing what is developing here. Okay, so in the case of intestine versus stomach epithelium, in the stomach, this BRX1 activates SFRPS. Where is that? Here. SFRPS, which then, so these are mesenchyme cells surrounding it, right? Let's take a step back. Okay. This is because this figure is kind of upside down, I think, with how they designed it. I didn't design this one. All right, so we have our endoderm, so our digestive tract. As it's proceeding through the body, its neighbors determine what it's going to turn into, right? So we have mesenchyme cells that are the neighbors. If these mesenchyme cells are expressing BRX1, then what's going to happen is these SR or SFRPs are activated, which then inhibit WNT. WNT always another one of our big players, right, that we talked about a lot in this class. So BRX1 through SFRPs inhibit WNT, which then leads the cells to turn, that part of the digestive tract to turn into stomach, okay? Now further down, if there is NKX2 and SOX9, that will form the sphincter in between your, so the gating, I guess is a good layman's term for it, the sphincter, the muscle or the tissue in between your stomach and your intestine, right? So that is there to hold the food in your stomach for a long enough amount of time to digest it enough to be passed on to your small intestine, right? So this is just more posterior of this developing tissue. Now on the other side, let's go back here. So the mesenchymal cells, so they're expressing BMPs, but in the absence of BARX1, what happens is there's nothing to stop WNT expression, and then it will just naturally turn into epithelium. So this intestine tissue, based on the mesenchymal cells being present and nearby, this is kind of the default, right? Is that WNT is just naturally expressed and it will turn into epithelium. Only in the stomach area, where the neighboring mesenchymal cells are slightly different in expressing BARX1, we get inhibition of this normal intestinal development and we get stomach instead. Okay. Let's see, any last notes? So like I said, in certain regions of the endoderm, cells excrete CMP4, which I don't think is on here, which instructs this SOX9 and NX or NKX2 transcription factors to form sphincter. So it's a little more detail into this, that a very small region of the endoderm, there's the CMP4, which causes the sphincter muscle. That's just for your information. It's not really on the thing here. After differentiation, the mesoderm and the endoderm continue to interact through development of this dorsal mesentery. And so this is a fibrous membrane that connects the endoderm to the body wall. And if this connection is severed, the mesentery shrinks and the gut becomes a long, thin tube with no folding. So there is kind of like we talk about this tissue interaction with like cadherins and things like that. The reason that your stomach is all, your guts are all contained and wrapped around in a specific pattern or a specific way is because of this dorsal mesentery, right? And so this fibrous membrane that connects your intestines to the anchoring points that need to be there to form that particular structure. So you can see in this case, if it is expressed, then we get this kind of like connective folded gut. And then if that is not present, that fibrous membrane or the dorsal mesentery, then we get just one long tube. So there is interaction or connection between the gut and the rest of the body. It's not just like this tube that sloshes all over the place in there. All right, that is it for this video. That is more on gut formation. Thank you for watching, and I'll catch you in the next one.